Hi, everyone. My name is Daniel Rickert, and today I'll be talking briefly about my summer 2022 research project, Conditional Coalescence Theory in a Population of Varying Size. So what exactly is coalescent theory? You may be familiar with the concept of a gene, strands of DNA or RNA that code for traits of organisms. In the simplest model, the genes of an individual are inherited directly from his parent or parents, with mutation possible in between parent and offspring. Coalescent theory is a mathematical study of the genealogical relation between genes in a population. For our research, we focused on a single gene that has two allelic types, say type 1 and type 2, that is, two different variants. We assumed that the population was large, essentially infinite, though varying in size throughout time. For example, the human population. We examined a genealogical relation among genes in a sample conditional on knowing the allelic types in the sample as well. It is helpful to visualize the relationship among genes in the population by drawing a coalescent tree, such as the one you can see here on the right. The tree structure is formed by tracing genes backwards in times through the parents of individuals and then their parents and so forth until we reach a single gene from which all genes in the sample descended, the most common recent ancestor. Sample types are represented by the larger red square and blue circles at the bottom, and mutation events, either a change to type 1 allele or type 2 allele, are represented by the smaller red squares or blue circles that fall on the tree. Looking at the previous figure, you may notice that the type of a gene in a sample is determined by the most recent mutation looking backwards in time. So we can form a new coalescent process, which we call the coalescent with killing, by terminating each branch when a mutation occurs, and we can then examine this process conditional on knowing the sample types, say N1 of type 1 and N2 of type 2. In this figure, we can see an example of the conditional coalescent process with killing. Notice how the type 1 individuals, red squares, cannot coalesce with the type 2 individuals, the blue squares. A quantity of interest, then, is the number of mutations of type 1 and type 2 in the process, which we denote by K1 and K2, respectively. In the constant population size case, it is known that K1 and K2 are independent random variables conditional on knowing the sample types. In fact, the entire sub-processes for the type 1 alleles and the type 2 alleles are independent of each other. Nonetheless, numerical calculations suggest that this independence is lost when the population varies in size over time. Nonetheless, there still appear to be some nice properties that appear to exist, even in this more general case. For instance, it appears that in at least some symmetrical cases, say conditional on having the same number of type 1 and type 2 genes in the population, and if we have some additional information about the timing of coalescence and mutation events, then K1, K2 are negatively correlated, regardless of how the population size is changing in time and the mutation rate. Furthermore, for certain nice population models characterized by an additional parameter beta, such as exponential growth or decay with rate beta, we can understand the rough behavior of the correlation of K1 and K2 as beta goes to 0 and beta goes to infinity. For instance, we know that the covariance of K1 and K2 is 0 to first order beta as beta goes to 0, and we have an analogous result as beta goes to infinity, and we suspect, based on a previous conjecture, that the covariance will be negative for all values of beta. With that, thank you for listening, and don't forget to check out the full project and extended slides available exclusively at lewisfanmath.com.